hello everybody. This is to welcome the ESID community to the first ESID Clinical Working Party webinar in the year 2023-24. On behalf of the head of the Clinical Working Party, Siobhan Burns, I welcome you to today's webinar, which has the topic of genetics. So we would like in this webinar to explain some difficulties in interpreting genetic results you're all receiving from your human geneticist colleagues. And we would like to discuss with you some difficult cases. And so you learn after this webinar to better interpret the results you're receiving from human genetics. And hopefully we will also cover some phenotypic and some classification issues. So you all know the agenda. We have wonderful speakers. I will now thank all of them before I forget at the very end that they have taken the time to think about presentation, to put this presentation together, to put a wonderful teaching session on. I'm looking forward to the presentations, and we would like to start with Dr. Andrés Caballero, who is giving us an introduction into the genetics and into the field. Andrés, the screen is yours.
Hello, everyone, and thank you for inviting me to give this quick presentation on the challenges of interpreting genetic reports for clinicians. So I'll start the first 5 minutes with this going over the basic nomenclature of human genetics. And for that, I'd like to quickly remind you something that might be very obvious for most of you, but so I just wanted to put it out there that we are diploid organisms. So that means that for every chromosome or gene in our genomes, we have two copies: one that we inherit from our father, and one that comes from our mother. So that's holds true for the 22 pair of chromosomes, non-sexual chromosomes. However, for the sex chromosomes, the X and Y, we males have only one X and one Y, whereas females have two X chromosomes. So why is this important? Because our diploidy is what determines or marks the different ways of transmitting traits and diseases. Here's a quick example. We have the most common modes of inheritance, so autosomal dominant recessive or X-linked dominant recessive for most of the known genetic conditions nowadays. In autosomal dominant, one parent, so either the father or the mother, is enough to carry and pass on the mutation and disease to the offspring. However, in autosomal recessive conditions, you need both parents to transmit the mutated allele and for the children or the offspring to develop the condition. However, in X-linked conditions, so usually the mother is the one that is transmitting the affected, so the mutated allele to the offspring. And in X-linked dominant, so either girls or boys can develop the condition, but in recessive, X-linked recessive, mostly boys are the ones that develop the condition, like probably most of you know, is the case of Bruton's A-gamma globulinemia with mutations in BTK, which is located on the X chromosome. But this is not that simple, like always in biology. So there are other possibilities. We have maternal mitochondrial inheritance, Y-linked conditions, mosaicism, incomplete penetrance, uniparental disomy, and so on. Just for you to have a general overview, there are about 30% of conditions follow an autosomal dominant way of transmission, whereas about 50% of them behave as an autosomal recessive conditions, and about almost 6% are X-linked conditions. So just putting the example of when we have one mutation in one allele, so this would be the case of the autosomal dominant conditions, in which the mutation goes from our gene, one copy of our gene, let's say in this case, in this example, the female, so the one that comes from our mother. So we have the mutation that goes onto the transcript, onto the mRNA transcript, and then it goes to the protein. So at the end, what we have is one functional copy of the protein and one mutated copy of the protein. In autosomal dominant conditions, having only one functional copy is not enough, and you would develop the disease. However, in autosomal recessive, you can live with that. So you can be a mutation carrier and not develop this condition. So this now, what I'm showing you now is probably what you will come across at some point if you're dealing with or working with genetics and doing genetic screening of your patients. Nowadays, most genetic screening is done via NGS, next generation sequencing, which could be using a targeted gene panel, a whole exome sequencing panel, or the whole genome. And basically, this would be what you would be looking at if you load the alignments of your results in this in a tool like the IGV. This is how an heterozygous or homozygous mutation look like. So meaning that if you see all these, I cannot use I don't see my mouse, sorry. If you see this blue bar completely blue, that means is it homozygous mutation. So basically it represents that the mutation is located in both alleles of the gene, so in both copies, the one that comes from the father and the one that comes from the mother. But if we have this bar that is half red, half blue, which is about 50%, that indicates that one mutated allele, so you have one mutated allele, which could be from the father or the mother, and one wild type allele. Now, just to let you know that there are some certain, probably many of you already know, but there are some certain guidelines or recommendations that were developed by the Human Genome Variation Society in order to put some structure to provide some framework in which you could describe this the mutations or the genetic variants, so that everyone understands how to report these mutations and how to interpret them. So here's an example. Maybe you would find you could find it like this on the right. So in the upper right, you would see the nomenclature at the chromosomic level. So if you receive a genetic report, they should tell you in which, so the coordinates that you see here, it should be either using the genome build. So there are different genome builds of the human genome. So you have the genome build GRCH37, the GRCH38, now the T2T. So they should tell you which reference build they use for annotating the mutation. And depending on that, the chromosomic location would look different. So what you see after this NC_0008, so that is chromosome 8, and the G stands for genome. And then you have the coordinate, a number that indicates the position within that chromosome, and then the change of T to A, that nucleotide. So there are usually not ways of referencing the location of the mutation at the gene level, but it's usually so reported in the transcript, so at the transcript level. And what you see here is this ENST, so this code ENST and a number, so that is the identifier of ensemble. So ensemble has a database of all the transcripts in genes in the genome, and that gives you the unique identifier of that transcript belonging to that specific gene, and it tells you what is the coding position. So so taking into account only the coding sequence of the gene. But you could also use the NM_, so that is from RefSeq, or you can even use the LRG_199. So that is also from the locus reference genome. So there are different ways of reporting the mutations at the transcript, at the coding sequence level. And you could also do the same at the protein level. So that single nucleotide variant would translate into a single amino acid change, and you would have different ways that, like put here, that you would find how they describe the mutation at the protein level. So here, just a very quick summary or overview of the most common type of mutations that are found or reported usually. So at the chromosomic or gene level, you can have a point mutation, single nucleotide variant, or SNV. So it's basically the replacement of one single nucleotide that can lead to different situations. So if you're close to a splice site, so in the exon-intron boundaries, this mutation gets altered the splicing of the mRNA transcript, which could lead to, for instance, the exon, so a skipping of one exon of that transcript. So you would have one copy with all exons of that gene, but one other copy of your transcript would not have that exon. So that could that skipping could then lead to an out of frame or in frame of so reading frame of the transcript, and therefore that could lead to a truncated or a shorter protein, which maybe in the end is degraded after all. So if we go to the other part, this a point mutation could also lead to in the end a missense mutation. So it's just this, as we saw in the slide before, a single substitution of one amino acid at the protein level. But we could also have some other type of mutation. So we can have insertions or deletions, which could in the end lead also to in-frame, frame-sit or nonsense mutations at the protein level, which all of them could also lead to protein degradation or even to nonsense-mediated decay, NMD. So the transcript would never get to be translated into protein. So just to now introduce the next part of the session, of the talk given by Adela, I would just like to just to introduce what I said in the slide before, that nowadays most of you would work with NGS, next generation sequencing data, and here's a short overview of the three different approaches compared. So if you do panel, so targeted gene panel versus whole exome sequencing versus whole genome, this is more or less what you capture. So with a panel, you would maybe capture a set of 200, 300, 400 genes. With whole exome, you would capture all the coding sequences of 20,000 genes. And with whole genome sequencing, you would capture everything: exon, introns, intergenic regions, everything. So yeah, of course, the amount of data generated with whole genome is much more vast and more complex to interpret and analyze. And here below just a comparison of what this would look, again, using a software like the IGV, what the whole genome sequencing would look like versus a whole exome sequencing or targeted gene panel. If we look at one gene, so ERC5 with 4 exons, so you see that for whole exome sequencing or targeted gene panel, you would only have covered that those pieces or those parts of the gene, which are the exons, whereas the introns, you cannot see anything. So would be a mystery. So with that, I'll go now with Adela. Leave you now with Adela. Thank you.
Thank you, Andrés. This was a very nice introduction in the field. And without further ado, I forward the microphone to Adela, who's going to deepen the topic.
So I will briefly discuss the importance of reanalyzing the datasets regularly. As you have observed in the clinic, the number of gene variants that are known to cause inborn errors of immunity have increased since 1990 significantly, and now we look at up to 500 genes in this group of disease. So what's the clinical implications of new gene discovery? Well, negative results might be caused by the fact that a gene function could be poorly understood or not at all associated with disease at the time of NGS analysis. So I'll briefly give an example on, this is a boy, 8 years old now, where we really struggled for a diagnosis and to understand his disease ever since infancy, because he had recurrent severe infections, both respiratory and urinary. And he was in an intensive care unit. He had pancytopenia, slightly low immunoglobulin levels, very low B lymphocytes. He had a bone marrow abnormality that was hypocellular. He also had additional features which were syndromic. He had motor delay, hypotonia, dysmorphic features, microcephaly. He also had joint hypermobility, eczema, thin, sparse hair, and enamel hypoplasia, abnormal tooth shape, pancreatic dysfunction. He had also growth hormone dysfunction deficiency, and retinal dystrophy and myopia. So many issues happened, and we really tried to understand what is the cause of all this. So we started with a classic karyotype, which was normal, and that had some fish for chromosomes 7, 8, and 22 that was normal. Array CGH was normal, male. We did some deoxybutan test for Fanconi anemia, which was negative for chromosomal breakage syndrome. A solo exome sequencing was performed in 2016 and was negative. This was done in another country, so the raw data was not available to our lab. Later, we sent for a panel because we wanted to look at deletion duplication for immune deficiencies. The panel was consisting of 207 genes back in 2019. So later we escalated to solo genome sequencing performed in 2020 from blood that identified 2 novel pathogenic variants in the gene DNA JC21. The thing is, both of these variants were exonic and could have been picked up in the exome sequencing. However, the association of the gene DNA JC21 was first done with the disease bone marrow syndrome type 3 in 2016, ironically the time of exome analysis. So take-home message, to be short, is that there is possible diagnostic gain of reanalyzing exome and genome data from patients with suspected primary immune deficiencies. And the reason for this gain is that you could have improved knowledge of genotype-phenotype correlation, you could have expanding the gene panel, and adding structural variants, deletion duplication analysis, which might not have been done at the time of first analysis. There are several publications showing the benefits of reanalyzing data from exome and genome. And very relevant to my talk today is an article published in 2018 where genome sequencing offers additional but limited clinical utility compared to reanalysis of whole exome sequencing. So do reanalyze your data or ask your geneticist to do so in case of a negative finding in a child where you really have a clinical suspicion. Thank you so much.
Wonderful. Thank you, Adela. So my topic is to talk about the importance of thorough phenotyping of cases to interpret the genetic results correctly. My case is the CVID patients, typical CVID, hypogammaglobulinemia, recurrent infections, upper respiratory, chronic diarrhea, and lymphoproliferation. However, only when it became clear that he had a sister who had an SLE-like phenotype and actually had died at age 16 following renal failure and a cerebral bleed, the interpretation of the exome became meaningful. Because in the beginning, we were looking for all those known CVID genes, but we had not taken into account other genes with more broad phenotypes, including the ones with causing interferonopathy. Of course, this patient, CVID patients, had ADA2. ADA2 was published in 2014 as a disease involving the immune system and the vessels, as a vasculopathy, or also as polyarteritis nodosa. Here you can see some pictures of patients. Many of them actually do also have involvement of the bone marrow and the immune system. Some of them with hypogammaglobulinemia, others with bone marrow failure syndromes. ADA2 can be also functionally tested. It's an enzyme, so you can send blood to test laboratories. For instance, Mike Hirschfield does it in the US, but there are now also many European laboratories who test for ADA2 activity. And you can see here that patients have an absolute absence of enzyme activity. And this is because the disease is autosomal recessive. So you need 2 defective alleles to be sick. That means if you have one healthy allele, like in parents, in heterozygous parents, they would have a retained ADA2 deficient activity, maybe not as high as normal, but sufficiently to stay healthy. When we confirmed the genetic results, we saw a compound heterozygosity. So what is that again? You inherit one mutation from the father. So this is the patient, he inherited one mutation from the father. The father's other allele was healthy, wild type. Whereas on that spot, the mother had a mutation. So the mother had a mutation at a different position in the same gene as the father, and the unfortunate patient had inherited the mutation from the father on one allele and the mutation from the mother on the other allele. So this is a combined heterozygous result, which made the patient sick because he had two defective alleles, but both parents are healthy because they have one retained healthy allele. Of course, when we did the phenotyping more thoroughly, we realized that the CVID patients had mild skin vasculitis at the lower legs, classified as livedo reticularis. So if we had communicated this feature of mild skin vasculitis earlier, we would have diagnosed him genetically much earlier, and that is really important because patients with ADA2 deficiency are at risk of developing strokes, cerebral strokes, and therefore they need to be treated, for instance, on anti-TNF treatment. Here's a summary with a nice publication from Johanna Schepp and Michele Proietti, summarizing all the phenotypic features you can identify in deficiency of ADA2, DADA2. If you're wondering what ADA2 is doing, we all know ADA1 from the SCID patients. ADA1 deficiency leads to SCID, and ADA1 catabolizes adenosine, and it's supposed that ADA2 does the same, and it's supposed that ADA2 also does that in serum, but it seems to be that it's not only doing that in serum, but also inside the cell, and this is data from Michele, who was supposed to hold this presentation in the beginning, but then I took over. Michele has shown that ADA2 is also expressed in tissue macrophages, as shown here, or in tingible body macrophages with other macrophages in the germinal center, where a lot of DNA is being captured by those macrophages. If you want to know what ADA2 is doing in those lysosomal structures of macrophages, you can read Michele's preprint in the bioarchive. So as a summary, there are phenotypic hints for specific genes within the large group of antibody deficient individuals. I talked about the CNS involvement and the vasculitis in DADA2, but vasculitis can also be present in patients with NF-κB1 mutations. Then if you see pancreatitis or rosacea or seronegative arthritis or hepatitis, you would need to think of NF-κB1 as an underlying genetic defect. If you have patients with enteropathy, that may also be NF-κB1, but that could also be CTLA4 or any T-regopathy where T regulatory T cells are affected, could also be CIAP. If you have autoimmune cytopenia, then CTLA4 or NF-κB1 again are high on your suspicious list. If you have alopecia, it's CTLA4 or NF-κB2 actually. If you have hormone autoimmunity or hormone gland autoimmunity, it's NF-κB2. If you see grammatose lesions, it's maybe CTLA4 or LRBA or any other Treg disease, and so on and so on. So these phenotypic cues are important to communicate to your human geneticist so they have an easier go to interpret the result. And this is my thank you slide. I'm doing the clinic together under the lead of Klaus Warnatz with a big team evaluating our patients and looking at them phenotypically in very much detail. Thank you very much. This was my contribution, and we save questions to the end. And the next presentation will be done by Roger.
Okay, thank you, Bodo. I will share my screen. So, okay, so this part is about a simple topic, which is essentially to keep in mind the novel phenotypes described for already known genes. So one of the challenges of our field is that novel genes causing inborn errors of immunity are constantly discovered. Ideally, you should keep in mind those novel genes, but our mind has a limited space, so at least keep it on your panel of IEI genes, either a virtual gene panel in case of whole exome or whole genome analysis, or if you have a targeted gene panel, keep it for the next update. But sometimes it is not a novel gene, but a novel phenotype that is described for an already described gene. And these novel phenotypes clearly expand the number of diseases caused by the primary immunodeficiency genes. So these novel phenotypes, which are sometimes quite different from the original descriptions, may lead to the genetic diagnosis of unsolved cases. And then you can do gene-focused reanalysis based on these novel phenotypes, even in targeted gene panels, because these targeted gene panels contain the gene due to the original description. So I will give you some classic examples and then some novel examples. As a classical example, most of you know the case of STAT1 that was discovered in 2003, the STAT1 deficiency due to autosomal recessive loss of function mutations, causing essentially mycobacterial and viral infections, and reflecting the role of STAT1 in the receptor of interferon gamma signaling pathway. And then 8 years later, a completely different phenotype was described due to autosomal dominant gain of function mutations in this case, restricted to chronic mucocutaneous candidiasis, and in this case, due to the impairment of Th17 immune response, which is crucial to control the candida fungal infection. Another example is the next member of a STAT, which is STAT2, that was described in 2013 as causing viral infections in humans due to its complete deficiency, so autosomal recessive loss of function mutations, and reflecting the role on the signaling through the interferon 1 and interferon 3 receptor signaling pathway. Several years later, in 2019, it was described the phenotype corresponding to gain of function mutations, in this case autosomal recessive, and these gain of function mutations produce an excessive amount of type 1 interferons and leading to a severe type 1 interferonopathy. So the both sides of the coin. And another classic example is STAT3 that was described by the group of Bodo, in fact, and others in 2007 as causing hyper IgE syndrome in an autosomal dominant fashion and causing recurrent bacterial infections, mainly in the skin and lungs, and other non-immune anomalies reflecting the role of STAT3 in IL-6 receptor signaling pathway and in other important non-immune signaling pathways. And 7 years later, it was described the phenotype corresponding to gain of function mutations of STAT3, and mainly associated to early onset multi-autoimmunity in patients. You can see here in this figure that both phenotypes are largely non-overlapping, except for the eczema and some dental anomalies. And this is other nice example about gain of function or loss of function mutations. The last classical example is complement factor I. I completely changed the genes to the complement genes. And I like this example because in this case, it is not question of gain of function or loss of function variants, but a difference between complete and partial deficiency. So complete factor I deficiency was described molecularly in 1996, although it was already described biochemically previously. The complete factor I deficiency, which is a regulatory molecule of the complement system, lead to a secondary C3 deficiency. And the clinical phenotype resembles, obviously, the C3 deficiency with recurrent encapsulated bacterial infections. And in 2004, occurred the first description of autosomal dominant loss of function mutation, so partial deficiency of factor I associated to a completely different clinical phenotype, which is atypical hemolytic uremic syndrome. This syndrome is caused by a dysregulated alternative complement pathway. There is a constitutive activation leading to this microangiopathic hemolytic anemia and all the renal diseases. So in this case, the complete deficiency and the partial deficiency led to completely different phenotypes. And I will show you now two recent examples of novel phenotypes of known genes that in our laboratory allowed the diagnosis of some patients that remain undiagnosed until that time. The first example is TRAF3. TRAF3 was originally described in 2010 as causing herpes simplex encephalitis, concordant with the role of TRAF3 in the signaling pathway through the toll-like receptor 3. This paper was a single patient study where they found a heterozygous de novo missense mutation, and they demonstrated an impairment of the induction of type 1 interferon through the toll-like receptor 3 receptor. After several years, it was noticed that this variant was not so rare. You can see here the frequency in NOMAD database. So probably this variant has a higher frequency to be a disease-causing. Maybe it's a low penetrance variant or even a risk factor, although it's functionally impairing some production of type 1 interferon. But the last year, the group of Taco Cupers and Kenneth Smith described different patients with autosomal dominant loss of function mutations in TRAF3, leading to TRAF3 haploinsufficiency. And in this case, the phenotype was related with B-cell defects, showing recurrent bacterial infections, dysgammaglobulinemia, and autoimmune disease, mainly Sjogren disease, celiac disease, vasculitis. And in this case, reflecting the role of TRAF3 as a constitutive negative regulator of NF-kappa B pathway. So when there is reduced TRAF3 in these patients, you can see here the different loss of function mutations. The reduced TRAF3 lead to a B-cell hyperactivation. In fact, most patients have this gamma-globulinemia in form of hypergammaglobulinemia, and this was a completely different phenotype compared with the previously described one. And the second example is regarding the otulin gene that was described in 2016 as causing early onset and potentially fatal autoinflammatory disorder in autosomal recessive loss of function mutation, so a complete deficiency. And in this case, otulin is a deubiquitinase that acts as a negative regulator also of the NF-kappa B signaling. And this systemic inflammation is led by the myeloid cells and by the TNF. You can see here that the complete deficiency of otulin impaired this deubiquitin activity and leading to this otulin-related autoinflammatory syndrome. So last year, in 2022, the group of Jean-Laurent and Casanova described another phenotype associated with otulin haploinsufficiency. So this example resembles the factor I example in the sense that the different phenotypes are due to the complete or partial deficiency of the gene. And what is funny is that in this study, they studied, in fact, the genetic basis of unexplained life-threatening staphylococcal disease. So they did an unbiased approach doing whole exome sequencing of more than 100 individuals. And they found an enrichment of rare variants in otulin gene. Otulin gene was the only gene reaching statistical significance. And the patients carrying loss of function, monoallelic loss of function mutations in otulin gene had extreme inflammatory responses to infections via Staphylococcus aureus, mainly manifested in the skin or in the lungs. They showed that the penetrance was not complete, in fact, quite low, 30%. And that defect was due to an impaired cell-intrinsic immunity in non-hematopoietic cells, so in fibroblasts, in skin cells, in lung cells, in response to this infection. And before ending, when I was preparing this presentation this weekend, I found another nice example published only 3 days ago that is related with the PLCG2 that was a gene clearly described in 2012 as causing an autoinflammatory disorder, or at least 2 autoinflammatory disorders called plight or aplight, due to gain of function mutations located in this protein domain. And just in September 13, so 3 or 4 days ago, the group of Jordan Orange and Anthony French described haploinsufficiency of PLCG2, so loss of function mutations associated to an immunodeficiency of NK cells, so NK cell deficiency. They described 3 patients of 2 unrelated families carrying monoallelic missense mutations that act as loss of function mutation. And this patient had normal NK cell numbers, but reduced NK cell killing. The patients preserved the B cell function that was impaired in the autoinflammatory disorder, and the partial knockout mice also displayed this impaired NK cell function. So it is another example of how we should keep in mind the novel phenotypes that are described of all genes, and that should be useful for future analysis or for reanalysis of unsolved patients. So thank you very much, and I will stop sharing my screen.
Thank you so much, Roger, for your presentation. Julianne, you are the next one to present your data.
Thank you very much for inviting me for this webinar. I will present you a case of finding Nemo. So, okay. The first case report is a patient who was a 9-year-old. It was a boy with a ID-like phenotype, including ectodermal dysplasia with sparse hair, abnormal conical teeth, and a slightly reduced number of sweat glands. Throughout life, he had an infection phenotype characterized by recurrent bacteremia and pulmonary infection with Streptococcus pneumoniae, Staphylococcus aureus, Pseudomonas, as well as disseminated infection with atypical Mycobacteria. The second family is a family history, a mother and her 2 daughters of panniculitis, uveitis, splenomegaly, and recurrent fever since birth for the 3 patients. And the younger daughter experienced sometimes arthritis. So laboratory investigation demonstrated low IgG and IgM and elevated IgA in the first case with a CD4 T-cell lymphopenia, and for the second case, an elevated C-reactive protein and a positive interferon signatures. So what is the suspected diagnosis? In the first family, we suspected an EDI with immunodeficiency, and in the second family, an autoinflammatory disease. So genetic sequencing was performed for both families. So for the first family, targeted EKB-KG sequencing was done, but no mutations were identified. Trio whole exome sequencing was not contributory. And in the second family, an head panel was no contributory, so the gene EKB-KG was not included in the panel at the time of the sequencing. And trio whole exome sequencing, so the trio is the mother, affected mother with unaffected parents, was not contributory. Finally, the genetic result was provided by full sequencing of the 3 uterine whole exome and most of the entries, so a full sequencing of EKB-KG gene in the first family, and by a trio whole genome sequencing in the second family. So what was this diagnosis? The result, as a result, we observe a branch point mutation in EKB-KG, NEMO gene. The mutation was localized in the intron 4 in position -23. So in the first case, it was A that comes to T, and in the second case, it was A that comes to C in the second case. And that explains why I presented you both family in parallel. So this is a well example of clinical diversity for a same mutation in the same gene. As you can see, the phenotypes are not matching. There is a history of conical teeth in the second family too, and a bit of sparse hair, but no really ectodermal dysplasia. And no history of recurrent fever in the first family. Moreover, about this mutation, you can see here that the routine in silico prediction for effect of splicing were poor. Splice, i.e., didn't find any effects and neither Maxent scan. So if you filter in routine with this algorithm, that is the prime algorithm used in whole genome sequencing filtering, you can miss this mutation. And fortunately, SPIP algorithm found the alteration of the branch points, and the effect predicted was exon 5 skipping. So mRNA studies were performed, and you can have more information about the first case in the publication of Trin Morgensen and her team in GACI in 2016. And we can see here that the EKBKG gene harbors non-translated exon 1 and coding exon 2 to 10. And the subsequent sequencing demonstrated the presence of NEMO lacking exon 5 in this patient, transcript lacking exon 3, 4, 5, and 6, and also transcript longer than the full-length NEMO transcripts, possibly representing missplicing of intron 4, generating transcript harboring truncated intron 4 stretches. So a complex translation of this gene, mutated gene. And in the second family, we are currently under processing the data with the Rafaela Goldbach-Mensky team at NIH, but it seems that only transcript lacking exon 5 is identified, but it's difficult to identify it in all the cells of the patient, and we have more families, so we would like to decipher a bit more the mechanisms of this pathophysiopathology, sorry. So this is unpublished now. Moreover, another challenge in sequencing EKB-KG gene is that there is a pseudogene that is called EKB-KGP1, and this pseudogene is located on the same part of the X chromosome, and it corresponds to the mirror duplication of exon 3 to 10 of EKB-KG. So you see here the EKB-KG canonical transcript, and here the mirror duplication of exon 3 to 10 of EKB-KG under the EKB-KG pseudogene 1. So this mirroring, this mirroring is challenging because one essential step in bioinformatic pipelines following DNA sequencing is to align each read correctly to the reference genome, and the presence of pseudogene can interfere with alignment, potentially leading to the detection of false positive variant mistakenly identified because they are mapped to the pseudogene instead of the gene, or a false negative variant not identified because they are mapped to the pseudogene instead of the gene. So the pipelines align the reads from the EKB-KG and EKB-KG-P1 locus to both regions, but this result is very poor mapping quality, and the presence of blank grid here means that the mapping is too poor to be included in the variant calling process. So you can see here that all the exons are white here. So this one is the exon 2 that is correctly mapped because it's not included in the mirror sequence of the EKB-KG-P1. And here exon 3, 4, 5, 6, and all these exons are in white, and that's because the mapping is not well done because the algorithm doesn't know if the sequence is to align to the pseudogene or to the gene. So you can see here an example. And moreover, the variant was absent of the VCF because it was this read were not included in the variant calling. And with a low allele valency, you can see here is 19% of allele valency for this variant because there are the gene and the pseudogene, so this dilutes the number of reads. Sequencing gene panel exome or genome is typically performed using short reads of approximately 150 base pair, and sequencing using short reads like this does not allow for the detection of the active gene from the pseudogene. Similarly, conventional Sanger sequencing is not feasible due to the very high sequence homology between the two sequences. So the main challenge lies in amplifying fragments of significant size and to include a missense between the gene and the pseudogene. There are only 6 missense in all the gene, including intronic sequences. You have to amplify fragments of size over 12 kilobase. So fortunately, there are some team that works, a team working in a reference laboratory that have provided a new method of detection of EKB-KG bypassing the EKB-KG P1 pseudogene. That seems very important to seek advice of reference laboratory when you have such difficulty, because they usually already have the protocols. So the take-home message of my presentation are to be aware of the filtering method when interpreting the results. So you have seen in this case that the splicing prediction weren't good. Be aware of the presence of pseudogene or homologous sequence. That's the case for the EKB-KG gene, but also for the NCF1 gene, or for ATD3A, there are homologous sequence because the MACPIM of some exons are 100% with 8AD3B or 8AD3C, CDC42, USP18, etc. Lots of genes are homologous sequence. And be aware of the clinical diversity, like Roger already says, and do not exclude potentially significant variants based solely on phenotypic issue.
Thank you.
Thank you very much.
Thanks, Jen. It's only in the interest of time that we quickly move on with the next presentation provided by Kimberly.
Thank you very much for having me. Let me put this on full presenter mode. So we're going to switch streams a little bit, and we're going to talk about expecting the unexpected and what happens when things don't go as you necessarily expect. So I'm going to quickly remind people about a slightly different disease. It's a familial hemophagocytic lymphohistiocytosis, and it's diagnosed either because of familial disease and a known genetic defect, or a number of very non-specific findings. So generally, you're considered to have HLH if you have 5 of the following 8. So fever, splenomegaly, cytopenias, and multiple lineages, raised triglycerides or ferritin, decreased fibrinogen, elevated soluble CD25, decreased or absent natural killer cell activity, and hemophagocytosis that might be on a bone marrow, it might be in another tissue like liver. And it's really important to exclude infection or malignancy, both of which can give you very similar phenotypes. And there's a number of different genetic causes of HLH. So these are the most common ones. So perforin causing FLH2, STXBP2 is the one I'm going to focus on today. That's also known as MUNC18-2 causing FLH5. But you can see there are other causes, and it's important to recognize that a number of other inborn errors of immunity, like XLP1 and XIAP, can mimic HLH. Their patients can present with classical HLH as in some of these other conditions. And a number of years ago, Jan Breshnan and Stefan L. and a group of us put together a flow chart that uses 5 mils of blood to try and rapidly diagnose these patients, because rapid diagnosis enables rapid treatment. And this is diagnosis, rather than being based on genetics, is based on functional assays or protein expression as well. So it's all based on 5 mils of blood. If the patients have albinism, you're going to do some hair microscopy and a blood smear. But otherwise, we look for the presence or absence of perforin. If it's present, we can go on and do other assays. If it's absent, they have a diagnosis of perforin deficiency or FLH2. If they're male, we'll go ahead and look at SAP and ZIAP expression. If absent, then they have XLP1 or ZIAP deficiency. And then we can look at NK degranulation. And if it's normal, then FLH is unlikely. If it's absent, it is likely. And there's some follow-on tests we can do if you get an indeterminate result or an intermediate result. But based on this flow chart, this is how we diagnose patients with HLH. And this is based on the fact that all of these signaling molecules work together. They're all involved in cytotoxic degranulation. So if you look here, you have a cytotoxic granule that can move along the microtubules. And as it moves along the microtubules, it can fuse with its membrane of the cell it's attacking and form a synaptic cleft and then kill the target cell. And in a very simple terms, this is what it looks like from an assay perspective. So the cytotoxic granules express molecule CD107A on them. That's intracellular, so you don't see it on the surface of the cell. When those cells are activated, the cytotoxic granules go and they fuse with the cell membrane. CD107 then becomes on the cell surface, and you can use an antibody to detect it. And as I mentioned before, this screens for FLH3, 4, 5, Shediac-Higashi disease, Griscelli's, Hermansky-Pudlak type 2. So we're going to go with the first of these cases. So we had a little 9-week-old girl. She presented to her local hospital with fever, splenomegaly, raised triglycerides, low fibrinogen, raised ferritin, pancytopenia, no hemophagocytosis on the first bone marrow that was done, but no viruses or other microorganisms are identified. There was no evidence of malignancy. Did she have HLH? Well, based on that, it was strongly suggestive of having HLH. Blood was sent to us. We undertook our assays. And if you look at this slide in the top right, you can see the flow cytometry plot for perforin expression. And this is an isotype control, and then this is her perforin. And I think you can see she has very normal perforin expression, making FLH2 quite unlikely. However, we then did a granular release assay. And these are unstimulated cells that have some background degranulation. And we happen to stimulate ours with PHA. You can see good degranulation. So we go from 13% to 30%. However, in our patient, very little background degranulation. And when we stimulate the cells, they didn't do much. They didn't go increase their degranulation. So we felt there was a defect. This was an abnormal granular release assay. So we could go on and we could do different proteins. So we did an immunoblot. And we did an immunoblot with a control, with one of her parents and the patient. And I think you can see this patient lacks MUNC18-2. There was no significant protein expression, although a control protein, actin, was present at normal levels. We did genetic sequencing, and she was homozygous for this mutation here, which was consistent with a diagnosis of FLH3. She had a transplant, was in our hospital for a while, got better, went home. Well and good. Good diagnosis, confirmed genetics, appropriate treatment. And then there was a second child. Second child, interestingly, was actually slightly older than she was. But a couple of years later, her brother, presented. Other than being male, they were very similar. They both had fevers. They both had splenomegaly. They both had raised triglycerides. They both had low fibrinogen. They both had raised ferritins. They had cytopenias. He had hemophagocytosis on the initial bone marrow. Interestingly, at 1 month, she did have hemophagocytosis on her marrow. No evidence of malignancy. We were not measuring soluble CD25 then, so I don't have those results. However, when we came to do his granular release assay, it was normal. But they're siblings, and she has FLH5. He must have FLH5. They're siblings. Come on. Same presentation. Look at it. Looks very, very similar. He did have some neurological symptoms. Quite common in HLH, nothing surprising. One clue, he had EBV. but so do lots of patients with FLH2 with perforin deficiency in the other forms. So not really that helpful. Rash, very nonspecific. He had some dysgamma-globulin anemia, but they were very elevated immunoglobulins, very consistent with an inflammatory pattern. So did our granular release assay not work? What happened? Did he have FLH5, mutations in STXBP2? Well, actually, he didn't. He had normal perforin expression. He had a normal granular release assay. These are samples that traveled in, so it's not the best flow cytometry, but I think you can see the control isotype in SAP. The control clearly expressed SAP protein. The patient clearly didn't express any SAP protein. And on analysis of his SH2D1A gene, had a confirmed mutation, proline 101 to threonine. So we have two siblings who presented almost identically. You know, on a clinical grounds, they both had classic HLH. You couldn't separate them out. But one had mutations in STXBP2, MUC18-2, and the other had mutations in SH2D1A, was an XLP. Treatment for the same at the moment, treatment for both conditions at this moment is the same. However, in the age of gene therapy, that may not always be true. And a genetic diagnosis may have you alter your treatment. Interestingly, if we go back to the parents, dad was a carrier of MUC18-2, not a surprise. Mom was a carrier of both XLP and MUC18-2, not a surprise. That's completely consistent. This sibling has never been tested, so maybe white's not the right color. It probably just needs a bit of shading. That sibling's never been tested to my knowledge. And mom's sister is also a carrier of XLP. Not a huge surprise. Everything's fine, makes sense. We go back the further generation to the grandparents, to the maternal grandparents. Grandmom carries the STXBP2, the FLH5 gene. Granddad, who died at age 42 of an atypical leukemia, almost certainly was the XLP carrier. Whether he was the index case, so we couldn't go back any further, or whether there were further family members affected, we don't know. But I think it's a really interesting story of expect the unexpected. Two siblings, same presentation, different genetic causes, and interestingly, very different genotype, no genotype-phenotype correlation, and that this child presented at age 4 with severe EBV-driven HLH, and this gentleman died at the age of 42 in the early 1990s of an atypical leukemia. So it's just a reminder that it's really easy. Most commonly, this would have been the same thing. That's what we see 98% of the time. But in a small percentage, something else is happening. So two diseases in the same family, or two diseases even in the same patient, which we have also seen. Sequencing for FLH or for any genetics, you do it for a variety of reasons. You do it for diagnosis, as in this case. You do it to confirm variants of unknown significance. Importantly, as I think a lot of the other people have spoken about, you may not find anything because the mutation isn't in the exomes or the region you're analyzing. It's not covered. You filtered it out. It's not a single genetic disease. And I really like this paper showing a impairment in the enhancer causing FLH3 that was missed by whole exome sequencing and filtered out by most whole genome sequencing pipelines. So just a really good example of some of what the other people have spoken about how important it is to both do reanalysis, but also to look at outside the regions, but mostly to expect the unexpected. This was obviously a team effort, so thank you. I'm sure we can take some questions in a minute, but I think there's one more speaker now. Thank you.
Yes, thank you, Kimberly. Wonderful presentation, wonderful case. We will finish off the round of presentations with Jonathan Bohlen from the Imagine Institute.
Hello, everyone. Thanks so much for having me. I'm a postdoctoral researcher at Institut Imagine in Paris in the Casanova research group. And today I have been asked, and I'm very happy to present to you some selected examples of very difficult to detect genetic variants and talk to you about what they are and how you could possibly find them. And I will also give you a small example of out of our own research work with such a case. So I think I really want to put the spotlight on genetic structural variants because they're really poorly discovered but very frequent and important type of variation. So these include large deletions or insertions, and these are basically copy number variations. They also include inversions or tandem duplications and also translocations. So even though these kind of structural variations are really underrepresented in a lot of the clinically diagnosed cases, they are actually expected to be, or if you analyze in an unbiased way, they make up around 25% of the predictive loss of function variants that are present in people. And so it is very likely that in our daily practice, we are missing a lot of structural variants. And so here I want to talk a little bit about how you can find them. So in the case that you know the locus where your variant is present, for example, you're doing family segregation, you can really use simple PCR-based methods. And these work very well, but the big disadvantage is that these are not a discovery tool, so you need to know what you're actually dealing with. High throughput sequencing methods such as whole exome sequencing and whole genome sequencing, particularly when they are short read sequencing, which basically all of them are, have really some problems to identify these variants. So in terms of the structural variants that I've listed, you can have some success in detecting copy number variations. There are several solutions for this, or let's say methods that have been published, including from our lab. You can and you should ask your genetics department to implement this kind of variant calling algorithms so that you can at least detect some of the copy number variations present in your patients. But for example, inversions are basically impossible to detect with these methods. And for this, you need more specific methods, such as the ones that I have listed here. Of course, the disadvantage with these is that they cost extra resources. They may not be available in your hospital or genetics department, but if they are available, and particularly if you have a patient with a strong phenotype that you are not able to find the genetic variant, you should suspect structural variants and you should try to use some of these methods to test this idea. Here is an example of a study from our lab where they have implemented such a method for the detection of copy number variation. This has been reported by Jeremy Rosin, who also helped me with the preparation of these slides. So a shout out to Jeremy. What you can see that there are several patients with IL-12 beta-1 mutations that are actually copy number variations. And so, for example, in patient 1, there is a large deletion removing a part of the gene. And I would say that this kind of variant, you can actually hope to be able to expect it just by visually looking at your exome trace. If you know roughly for which gene you are looking for, this is visible. But then in these other patients, you have this kind of large deletions in a compound heterozygous state with a missense or a peel-off variant. And this is going to be very, very hard to detect by eye. And you need this kind of variant calling algorithms to detect a difference in sequencing depth, as, for example, observed here. Okay, another type of mutation that has been actually also briefly mentioned by a previous speaker that I want to talk a little bit about because I think they don't get a lot of attention are branch point mutations. So these are mutations that impair the splicing of the pre-mRNA that is synthesized from the gene that is affected. If you don't know, the branch point is a sequence pattern that is present somewhere in the intron between two exons. So probably you are aware of the splicing consensus sequences that are at the intron exon junctions, and these are part of the routine variant calling that all of us use. But there's also a very important sequence motif that is the branch point, and that is inside the intron. And that means one problem is already that all of the whole exome sequencing don't cover this area. Yet, if you have whole genome sequencing data, then I suggest you ask your genetics department to use the method published by Pang in our lab last year to try to identify some branch point mutations in those whole genome datasets, because mutations in the branch point can easily be loss of function mutations, just like mutations in the consensus splicing donor or acceptor sites, and they can lead to completely aberrant splicing and loss of your genes function. Finally, I want to very briefly talk about somatic mosaic mutation. So these are mutations that are not inherited, but they occur at some point during the development or even during the life of the patients. These are excessively difficult to detect by whole exome or whole sequencing data, particularly depending on the on the allelic burden that is present in the tissue that you are sequencing DNA from. And so generally, single nucleotide somatic mosaic mutations are currently not easily detected by our methods that we commonly use. It would require a very high coverage sequencing that is also very expensive. I think in some cases, if you have a familial segregation that is not adding up because your patient carries a pathogenic variant but is not present in the parents, that's when you can, infer that it's a de novo somatic mutation and it's likely mosaic. Or if you have a strong gene suspicion, I do encourage you to look at the trace of this gene in your genome viewer and see whether there are any heterozygous variants that have a really skewed allele ratio, that they are not 50-50, particularly if they are covered by a lot of reads. And in this way, you could detect some somatic mutations. But generally, single nucleotide mosaic variants are very difficult to find. Yet, there is a type of somatic mutation that can be detected. It's a method that we are currently developing. And what I mean is somatic uniparental isodisomy. And so somatic or uniparental isodisomy is the phenomenon when a cell or a whole organism inherits 2 copies of the chromosome from the same parent and no copy from the other parent. And so this leads to a copy number neutral stretch of homozygosity in the genome, which can underlie recessive genetic disease. And here are some examples how this uniparental isodisomy can occur. In the embryonal stages, it can occur as a trisomy or monosomy rescue, and this leads to whole chromosome isodisomy. But also in the complete life of an organism, this can happen at any point in time as a mitotic crossing over event. And in this case, the isodisomy is typically segmental. And in fact, there aren't any methods yet available to detect this in a comprehensive manner in whole exome sequencing data, and it's something that we are working on. So I want to present you one such case that we are currently investigating. This case is a French family that has monozygotic triplets. So these are 3 boys that are, let's say, genetically identical, and they have presented with recurrent pneumonia. And now at the age of 10, they have parenchymal lesions in the lung that resemble emphysema. And it has been a really long time that our lab has looked into the exomes of these patients until I identified a homozygous missense mutation in the CBL gene that is actually already reported to be pathogenic, but in a completely different disease. And when I did the familial segregation, this was really surprising because only the father is a carrier of this missense mutation. And so the explanation came from a careful inspection of the allele ratio of the variants on the chromosome 11 on which CBL is located. And what we observe is exactly this stretch of homozygosity that is a result of uniparental isodisomy. So this observation led us to the conclusion that these patients inherited 1 copy of the CBL missense mutated allele, and then at some point in their development, a uniparental isodisomy on a segment of chromosome 11 occurred, leading some of their cells to be homozygous for this mutation. And indeed, when I genotyped different tissues of the patients, we observed that hematopoietic cells are highly have a high mutational burden for this variant, whereas non-hematopoietic tissues are heterozygous. And so this confirms that it's a mosaic mutation. And even within leukocyte subsets, we can see interesting differences in the allele burden. And this is something I am currently investigating to explain different types of phenotypes between these different patients with CBL mutation and uniparental isodisomy. Just a little bit clinical background. You may know that CBL mutation underlies a predisposition to develop Noonan syndrome, so developmental defects, and to develop juvenile monomyelocytic leukemia. This juvenile monomyelocytic leukemia is typically triggered by a loss of heterozygosity via uniparental isodisomy in the leukemic cells. And what has been reported is that even after complete remission of this leukemia, the leukocytes of these patients remain homozygous. And so what we are investigating, and this is an ongoing project, is several of patients with known CBL pathogenic variants that do not present the classical phenotype. So 4 of these patients had severe and life-threatening bacterial infections. Several of these patients have autoinflammatory and even autoimmune problems that have already been reported in the context of this CBL syndrome. And all of them, whether they had a leukemia or not, are homozygous for this CBL mutation in the blood by uniparental isodisomy. With that, I hope you could take away some lessons from this talk. I want to thank everybody who's involved in this research. I want to thank Jeremy, and I look forward to answer some questions.
Wonderful, Jonathan. Thank you for this very nice presentation. So I was actually thrilled by listening to all those contributions. They were really spot on the top point, and I'm now looking forward to the questions from the audience. Please put them into the question and F&A session question F&A button, and there have already some questions being listed. I think there are 7 right now. We have another 16 minutes to answer them. I will read them and then ask people to respond. Maybe I will take the first question. Sometimes we see heterozygous variants that correlates clinically to an autosomal recessive immune-mediated disease. Why do you think this is possible, and could it be that the genetic tests have failed to detect the other allele? So first of all, I have to say this is not sometimes that we see it. It's quite often that we see heterozygous variants in individuals, heterozygous variants of genes which are known to have an autosomal recessive trait. That is then often not likely the cause for the disease. Yes, of course, it is possible that you have missed the second allele, a mutation on the allele. We have talked that it's not easy to identify branch mutations. It's not easy to identify all different splice site mutations. So yes, you want to definitely look at this gene in detail. But yes, these diseases are autosomal recessive because often if there's only one mutation, it doesn't lead to disease. And therefore, these heterozygous variants are detectable in the healthy general population. The next question is about, we get a lot of variants in different genes, which may or may not be directly linked to particular immune-mediated disease genes. How should we approach those cases where the stronger variants are identified in genes with partial overlap with immune-relevant genes? Who wants to take this question? Andrés, what is your approach if you see a lot of variants in genes which are not definite part of the 500 immune relevant genes? Do you report them or don't you report them?
Yeah, I guess that depends on the lab, or I mean, there's also this list of genes in which you should or you're allowed to report incidental findings, or so if you find those, so I think there's I don't know if some of you has the link now, but I think probably in the NCVI website, so you have a list of genes in which you would be allowed to report them, but that depends. I mean, I guess different. So if we're speaking about a research lab, so maybe you report more than that you should, or yeah, I guess it really comes down to the lab or your own guidelines.
Guilaine, how you're doing it in France?
It depends if the variant explains the phenotypes or not. We report the variants that are explaining the phenotypes only. And the other are incidental findings. So,
and the second question of this listener is, if you have a very strong clinical and immunological evidence for a particular disorder, but you identify only benign or likely benign variant in genes of choice, should you consider functional testing? My answer would be definitely yes, because you cannot rely on those predictions. So it depends a little bit where you have, on what database you're looking, and what algorithm you're using for the prediction. But definitely the prediction programs are not very well, and you cannot rely on. So definitely functional testing is really important. Any other comments regarding this question from the panel?
yeah, just if the classification is based on the allele frequency and GNOME ID, and the frequency is 50% of the population, I think it's not needed to test the variant.
I agree with you. Gian, what is the cutoff? You're saying 50% allele frequency, definitely no need to functional test. But where is your cutoff? Let's say we talk about CVID as a phenotype, which has approximately a prevalence of 1 in 10,000 in the population. So is it 1% in the general population, or where do you take the cutoff?
The SCMG cutoff is 5%.
5%.
Standalone, you know.
Mm-hmm. That's interesting. The next question may also go to the more human genetics-oriented people, asking about our experience regarding the variants likely benign using HMTG criteria guidelines, conflicting results on ClinVar, and would it fit the patient's phenotype? Would it be reported or ignored? So I guess, conflicting results on the computer prediction program is no reason at all, because they are often wrong. What would you say about ClinVar and the American human genetics guidelines?
Now, at this time, ClinVar is not curated, so there are real good evidence in ClinVar, and also lots of bullshit, or if it's not
well curated. I agree.
Why it's complicated to interpret.
Yeah. So especially for the immune relevant genes, I think Michele and Andres have found that the ClinVar data is not very helpful, and therefore, this GENIA database approach has been started within ESID to better annotate immune relevant genes. So I think it's better to report than to ignore. And definitely functional testing is also here the response to go. Then there are two questions. Oh no, one comment and one question to Kimberly. Did the mother of the two HLH patients also have HLH?
No, so she's a carrier of XLP, and we know most of those carriers don't manifest with XLP unless they've extremely skewed exon activation. And she's a carrier of STXBP2, but again, most carriers remain well through their life. So at the moment, it doesn't mean she always will be, but she's in her 30s and well.
Thanks. So I guess the next question also goes towards you. The next two questions, actually, Kimberly. The first one is a management question. It has nothing to do with genetics, but it asks about a boy who's presenting with hypogamma and EBV infection, but no HLH, on immunoglobulin replacement, SAP mutation detected, but no access to gene therapy. Would you suggest bone marrow transplantation or just follow up?
I think we would tend to transplant most of our XLP1s. I don't know what other people do, but I think most of them we would transplant, particularly if there was a good donor available. I think eventually they run into trouble, and I don't think we have any long-lived XLPs, to my knowledge, on the ESID registry or anywhere else. I mean, they can clearly make it into their 30s, but with compromised quality of life. I don't know.
And
what
that's due to risk of developing HLH in the future or lymphoma?
Well, both. They can get lymphoma, HLH, they become aplastic, they can get vasculitis. That we had one unfortunate child who got a cerebral vasculitis. I think they can get so many things that are not compatible with high quality of life.
Another Kimberly question. Isn't it rather unusual to find both autosomal recessive and X-linked mutations causing the same rare condition? So basically raising a question whether this family is possibly consanguineous and we don't know. Or is it just bad luck?
Well, they're almost certainly unknowingly consanguineous because it's the same mutation on both alleles. So it's a homozygous autosomal recessive mutation. It's not a compound heterozygous. They come from the same relatively small village, and it is probably some form of a founder effect for the STXBP2. I think for the SH2D1A mutation, the XLP mutation, we don't have that evidence, and we don't know how far it goes back, and it could have been a spontaneous mutation in the grandfather.
Next question goes to Adela. How often do you recommend to reanalyze genetic data sets for unresolved cases?
So it really depends on your bioinformatic team. There are tools that do this in an automatic way, or you could have a PhD or two or a few that do this, or it can actually be something that a clinician could request to the team. So they see the patient in the clinic and then think, okay, this is unsolved, we don't know what it is, or there's phenotypic new clinical issues with the child.
I agree,
So
and it also depends.
It's very useful to have new data from the phenotype to ask for reanalysis. It's very useful.
I agree, and it also depends a bit on the new treatment opportunities. So if we now realize that there's specific treatment available for CVID patients, we need to reanalyze our CVID cases to make sure that there are not more ADA2s amongst them. You can treat with anti-TNF, or more CTLA4s you can treat with abatacept, or more NF-κB1s you can hopefully also treat in the future better. So I guess it depends a little bit on the phenotype, whether you think there may be specific treatment available. Then there's another HLH question to Kimberly. You've answered the first one already, regarding the mother clinical presentation. But how was her CD107A degranulation assay?
She had a normal degranulation assay, and she expressed relatively normal amounts of MUC18.2 protein.
Thanks. Now, Jonathan, back to you. Besides the CBL missense mutation, do you find any other pathogenic variants in the homozygous region? How large is the uniparental disomy region in your patients?
Hi, thanks for the question. Actually, I think this is Xinhua, my colleague, so from China. So great to hear from you. The region is really impressively large. So in the couple of patients that we have analyzed, it's between 50 and 80 megabase pairs. So it's almost a half of the chromosome, and it encompasses around 400 genes, depending on the patient. In these patients that I have analyzed, there are no other mutations that are predicted to be pathogenic, but it's very likely, let's say it's very possible that there are other homozygous mutations that could cause some phenotypes in some patients. In this particular project, we are relatively sure that the common phenotypes that we see are caused by the CBL mutation, because this is really common to the now 9 patients that we are studying, and they actually all show similar cellular phenotypes, which is also something that we are excited to report at some point, so that people can use this cellular phenotype to do a functional test. Thank you for the question.
Thanks, Jonathan. Next one, Roger, maybe you can comment on, I have 2 siblings with immune deficiency, whole exome sequencing has failed to identify common variants. Next step, need help.
Yeah, there are different possible next steps. One, it could be whole genome sequencing. In this case, I would recommend also include parents, because the number of variants that you will obtain is huge. So to analyze the different modes of inheritance, it's useful to have affected patients, but also the parents, to filter for different modes of inheritance. Then other options may be related with the talk of Jonathan, doing some kind of specific techniques for structural variants that may be missed in whole exome sequencing, and in some cases even in whole genome sequencing. That's the general answer about that.
And I have to add that we in Freiburg still have families, mother and daughter, 2 siblings. We have quite a few, at least 5 families, which despite doing all that, it's still unresolved. So you're not alone, anonymer Zuschauer. So you're not alone, but please reach out to the people on the webinar for collaboration. They are all very experienced. So you can go to either one and ask for collaboration. I think that would be really helpful. Next question from Elizabeth. I find a variant to be uncertain, but show that functionally everything is normal. Then the variant can be classified as non-pathogenic. And if so, there's an online database where it can be reported, so the other research or clinicians would find who I find it will know. I quickly answer because you have to be careful. For instance, with NF-κB1, there are variants which are novel. Manfred runs 5 different tests, from Western blotting all to reporter assays, and they're all normal. But, for instance, he does not look into the dimerization between NF-κB1 and P65. So there are functional things we do not test, even though we run 5 different functional tests. So it could be that this mutation impairs the dimerization of 2 proteins you don't even know. So I would say that you can classify it possibly non-pathogenic, possibly benign, but if your functional test is negative, you cannot conclude that it's definitely benign. There is a new database coming up. I was quickly mentioning previously, GenEA. So it's Andres help me, Genia, www.genia.db, then geniadb.net. Okay, geniadb.net. And in that database, we need curators for specific genes where you can then add your functional testing so everybody can look it up in the future. That shall become the new OMIM for immune-mediated diseases. Now we are approaching 5:30, and I would need to close down the webinar soon, but not without saying that maybe people on the call can answer the remaining questions directly. If you push the blue button, respond, you can respond to the questions you think which go towards your expertise. And please also, you can also answer, several people can also answer. And I do want to give the last word to Eleanor, first with a big thank you for organizing this wonderful webinar. It was very smooth running. Thank you for giving us the opportunity and for announcing, hopefully, the next ESID Clinical Working Party webinar. Eleanor, back to you.
Thanks very much, Bodo. And I'd like to thank again all of the speakers for their wonderful presentations and for participating, and also to Bodo for doing such a great job moderating this session. And of course, to all of our wonderful participants and for your insightful questions. Just a couple of technical points for future sessions. So a recording of this session will be made available within 2 weeks on the ESID YouTube channel, so keep your eyes open for that. We'd also like to give a special thank you to the sponsor of this session and also the next couple of sessions throughout this year and early into 2024 of the Grand Rounds, which is Barcelona PID Foundation. So thank you so much. Your sponsorship is really appreciated. And the next session will take place in October of the Grand Rounds. We don't have a specific date, but that will be announced very soon. So keep a watch on this space. The topic of the next session will be flow cytometry in PID. So thank you again, everybody.
Thank you. Bye-bye.